Evolution is the changing of the alleles in a population over time. Laws are things that occur. Theories explain laws, and theories also are used to make predictions. One of the predictions that has happened in the past is the prediction of how life came about. And this is something that we will get into when we talk about abiogenesis. Today we're going to talk about endosymbiosis. Now, when you look way back into the history of the earth, you find that the very, very old rocks have examples of very primitive life forms. The rocks that are very, very old are bluish in color if they had iron in them, and the reason for this is because they didn't have oxygen, and oxygen oxidizes the iron into a red color that we would call rust. As you get higher and higher, you see that the life forms apparently get a little bit more complex, and they get more in numbers, and they evolve the ability to release oxygen. Obviously, photosynthesis has evolved into the life forms. So now we have things that are photosynthesizing. We call those the cyanobacteria, or blue-green bacteria. Now, bacteria come in two forms. For a long time, we didn't really worry about it. We just called them all Monera, Kingdom Monera. But now the Kingdom Monera has been divided into two different groups of bacteria. They are called the Archaeobacteria and the Eubacteria. Archaea meaning very old, and Eubacteria meaning true bacteria. When you look at the Archaeobacteria, you find that the Archaea actually function with their RNA the way that modern eukaryotes do. So there are three different groups of living things. There are the Archaea, the Eubacteria, and the Eukarya. The Eukarya are the ones that have a true nucleus, and the other two are grouped under the idea of prokaryotes, which means they are before there were nuclei. But to call something a prokaryote means that it has no intracellular membrane-bound organelles, not just nuclei. When you look at them, you find that, as I said, the Archaea function the same way as the eukaryotes, and the eubacteria have a different type of RNA. They put the proteins together differently, and so they function differently from what the others. In fact, when we do genetic engineering into eubacteria, we have to find the RNA, we have to do reverse transcription, and then we have to insert the DNA directly because it doesn't chop out the different parts, the introns, and put the exons back together to read them. So we have to do it ourselves, which we do. That's how we are able to genetically engineer bacteria to create human proteins. And we do that too. That's the reason we have human insulin that is being manufactured by bacteria. Well, when you look at these things, you find that as time went on, you started having the red rocks, meaning that now, as you move higher up in the layering of the earth, you have the redder rocks where the iron is there, meaning that oxygen has been provided. Oxygen is the end receiver of the electron transport chain in respiration, and as the end receiver of the electrons, it allows the production of an excessive amount of energy from every sugar molecule, whereas if you don't use oxygen, you end up with basically fermentation, which is how we get alcohol and vinegar, and the reason your muscles hurt whenever you overexert, because you don't get all of it and you end up with the waste products. But with oxygen present, you can get all of it used up, so you don't have alcohol production if you bubble oxygen through the mixture. You end up using it up and turning it into carbon dioxide and water, the same as what you do when you basically burn things up. Well, it wasn't always that way. At some point in time, you ended up getting things to work together. And a lady named Lynn Margulis was looking at cells, eukaryotic cells, and she noticed that the mitochondria had two membranes. Now, we can tell lots of things with the transmission electron microscopes, and when they look, they can find out all kinds of things about them, especially when they put them together and regenerate what they look like with the computer. And the discovery was made that the mitochondria had two membranes, and they had a layer that was all wrinkled up on the inside called cristae, and that on this membrane you had the process of electron transportation and the breakdown of the carbon hydrogen molecules to release the energy that is necessary for a lot of energy production. So the mitochondria were labeled the powerhouses of the cell. Well, they looked also at the chloroplasts, and sure enough, the chloroplasts had two membranes. One of them was arranged differently into what were called grana on the inside, and in these grana you had photosynthesis occurring. That's where you had chlorophyll. You also had the electron transport chain in photosynthesis, and you also had the inside where the hydrogens were being placed and pumped back across, and you had as much energy as possible being extracted because of that inner membrane. But the question was, why do these things have two membranes? And Lynn Margulis came up with the answer. She said, listen, these were once free-living bacteria. They now live inside eukaryotic cells as organelles. Now, the definition of a eukaryotic cell is that it does have intracellular membrane-bound organelles. And she said that's where these things came from. And sure enough, look at them, find out both mitochondria and chloroplast have their own DNA, and both mitochondria and chloroplast divide independently of the cell. And that other membrane was when they were taken in by endocytosis, transported inside. Obviously, a cell at one time attempted to consume the other cells and couldn't consume them. Now, we know that rickettsia today function very similarly as the mitochondria, and we know that the blue-green algae, some of them are almost identical to the chloroplasts. And so all you have to do is kind of put it together and do some checking and find out, yes, these things are the descendants of what were once free-living bacteria. In fact, our cells, animal cells, are combinations of at least one former free-living bacterial cell that got in to become our mitochondria, and another type of cell which apparently was archaea eating eubacteria because they function differently. We know also that the nucleus itself has a membrane that is envelope, they call it, that's double layered, and there's a possibility it also had origins that were separate, but it contains now the DNA. The mitochondria have their own DNA, and most of the cells of your body, most of the DNA in the cells of your body is actually mitochondrial DNA. Without the mitochondria, you wouldn't be able to survive, you wouldn't be able to be multicellular. The huge monster that you are could not exist because you wouldn't have the mitochondria to provide the energy to be able to build this huge colony that you are. It's because of the endosymbiosis, bacteria living within other cells, that you are able to be what you are. So when people complain and say that they're not the product of evolution, they're not only the product of evolution. Evolution is taking place two places in their body, in their mitochondria and also in the nucleus. You have the transfer from generation to generation to generation. And in fact, the way we tell when you had a common ancestral mother with other humans, which I said it wrong before, was probably about 300,000 years ago, that all of us humans shared a common ancestral mother. We can tell that by looking at the mitochondrial DNA and looking at the regular mutation rate that occurs in these things and trace it back to when there was a common ancestral mother. We can also tell when any one of us shared an ancestral mother, even not back to the original. But we can trace all humans back to one ancestral mother 300,000 years or so ago. Of course, by the regular DNA, the Y chromosome that's found inside the nucleus, we can only trace ancestral fathers back to about 100,000 years ago, which means the two were quite separate from one another in time. They had a big temporal isolation. So Adam and Eve didn't live in the same time period. They were separated by lots and lots of years. At least the genetic ancestors. When you look at this, then, you find out that endosymbiosis is a remarkable thing. And when you look at the plants, it's even had an additional thing, which now they have two endosymbionts. The endosymbiont of the mitochondria and the endosymbiont of chloroplast. And they're different. They function with bacterial DNA. That means they have bacterial ribosomes, and bacterial ribosomes are not membrane bound, but they are organelles and they function differently than the ribosomes of archaea or of eukaryotes. Consequently, you can literally take things to kill out the intracellular bacteria, and so you can actually kill out chloroplast and leave the plant cells alive, causing white plants, just by putting something in to work against the ribosomal RNA. Now, ribosomes, you know, are what put together the proteins, and ribosomes, as you know from, well, you're going to find out the next session, ribosomes actually function as enzymes themselves. So they actually carry out processes and they can actually put things together and make things work. So you can kind of put two very primitive organisms together, the archaea and the eubacteria, and you can bring as a combination the organisms into the huge colonies that we have today. And even though all of the cells in your body are a part of the same colony, they are each an individual life form. Every cell is an individual living unit. You are a colony. You are a colony that specific parts are involved in reproduction and specific parts are involved in thinking. As such, the part that is involved in reproduction goes through a different process than the thinking part. And as such, any individual cell is a living part of your colony with the same DNA and the same mitochondrial DNA and lots of mitochondrial DNA, because all of the mitochondrial DNA comes from your mother. So when you have an individual cell, when you kill it, you're killing an individual of the colony. The same as if you have a fertilization event. If you kill that fertilization event, you are killing an individual cell. You're not killing an individual. You are the product of a huge colony that all has to be there to be able to function. Any individual unit is just a unit of the colony. Remember, populations evolve, and if you're going to call yourself evolving, you have to consider the whole population evolving, and it's quite obvious you evolve, you get older, and then you die. But populations of individual colonies evolve in living things. And that's what we talk about as evolution. Evolution is the changing of the genes, the alleles in a population. And I'm not talking about the mitochondrial DNA, but it can evolve also within the colonies. The changing of the genes in a population over time is evolution. The laws are the things that occur, and change always occurs. That's a law. And theories explain the laws, and theories are the things that can be used to predict. That's the reason we can predict what is going on evolutionarily, and we can definitely make predictions, and they happen.